So, hello students, welcome to CBSE NCERT Solutions. This is Basraj, your biology master teacher. Now, on this channel, we find solution to given every single question in your NCERT. At the same time, we also do NCERT line by line to make sure you do not miss out any point given your NCERT. Now, the topic for today, our chapter for today is respiration plants. Before we start today's class, let me tell you the weightage of this chapter. Right. Now, this chapter right here, respiration plants, usually there are around 4 to 6 questions are generally asked from this chapter. 4 to 6 questions. And on an average of 5 questions will always be there from this particular chapter. 5 questions. And all of these 5 questions are going to be from either from 1 or 2 questions from glycolysis, 1 question from electron transport chain, 1 question from or 1 or 2 questions from Krebs cycle, 1 or 2 questions from balance sheet, amphiboic pathway. There are multiple areas where question can be asked from. And let me tell you, students, each and every single section is equally important. So I want everyone to pay extra attention to every single line of this chapter. Okay. So with that being said, let's start today's class, respiration in plants. Now, all of you. Now let me ask you a simple question first of all. What is the difference between breathing and respiration? Let's see how many of you can tell me the difference between breathing and respiration. Now, all of you will tell me, sir, breathing is a much more physical process. Breathe in, breathe out. So, breathing is a much more physical process, while at the same time, respiration is more of a chemical process. Respiration right here is more of a chemical process altogether. Chemical process. Now, what do I mean by chemical process? It is a chemical process in which a lot of reactions are taking place. It is technically also called as physico-chemical process. Okay. Now, I will ask you one more question. Now, breathing. Where does breathing takes place? You will tell me, sir, breathing takes place in the case of your lungs, trachea, and everything, and especially the nose, nostrils, everything play a major role. Now, what about respiration? Where does the respiration takes place? You are supposed to tell me, sir, respiration is a process which takes place inside a cell. That's right. Inside every cell, there is respiration. It could be aerobic, anaerobic, and everything is there. Okay. So it takes place inside a cell. Now that point is clear to all of you. Difference between breathing on one side, respiration on one side. Okay. Now the next other question is, I'm talking right now, blah blah blah, blabbering. Okay. Now this talking or working, walking, eating, sleeping, any physical process or any process, does it require energy? Obviously. For me to talk in front of you, for you to listen and concentrate, every single aspect will require energy. हर एक चीज के लिए एनर्जी जरूरी होता है, राइट? सो एनर्जी जरूरी होता है, सो एनर्जी चाहिए, राइट? वी नीड एनर्जी. नाउ वेयर डज दिस एनर्जी कम फ्रॉम? लाइक आर वी बाइंग द एनर्जी फ्रॉम समवेयर? नो. Is it sold in Swiggy and Zomato? Yes, technically yes. But what is this entire energy part? Or where do we get this energy from, friends? So the energy is always and always obtained from the food which we eat. That's right. The food which we eat, nice chicken biryani, or your pizza, burger, anna sambar, rice and sambar, right? Chapati and palya, all the food items there, no, that will give you the energy. Right? That will give you the energy. But students, if I ask you, where do we actually get the energy from? For example, this entire biosphere. In this entire biosphere, where is the primary source of all of this energy? See, yes, rice we are getting from market, but how is the rice produced? We are eating chapati. Chapati is made of wheat, ragi, or jawar, anything. But where are we getting all of these items from? We are eating chicken biryani. Chicken is eating plants. So, entire primary source of energy, can I call it the ultimate plants? Tell me, sir, yes. The ultimate food source is always the plants. Plants. It could include your monocots, dicots, algae, bryophytes, pteridophytes, gymnosperms, angiosperms, name it. But plants are the ultimate source of energy for every single organism to survive. Except some chemisynthetic bacteria, there's a different story altogether. Okay, so plants give us the energy. But how are the plants giving us energy, students? Is the plants going and buying from the supermarket 1 kg energy? No. Plants can prepare and make their own food. And the process of making the own food is called as what? It is called as your photosynthesis. So in the previous chapter, that is photosynthesis in higher plants, we learned in detail about this. We learned about light reaction, C3 cycle, C4 cycle, C2 cycle, chemosynthetic hypothesis. Every single step we learned in the previous chapter. So please remember that whatever energy we need, right, it's coming from the plants. It is coming from the plants. Okay, photosynthesis. And whenever this energy is given, so we are getting the food now, right? The food is here. This is a primary source of food we have now. Now this food right here, this food will break down in the presence of oxygen or absence of oxygen, or this food can get oxidized. Right? Food can get oxidized. What is oxidation? Loss of electrons or loss of protons. This food can get oxidized to produce energy. To produce energy. And this energy is stored in the form of ATP, adenosine triphosphate. This process here, from the food to making energy, is called as respiration. It is called as respiration. Let me tell you one more time. Let me tell you. Listen very carefully. For example, this is your human cell. This is your cell. Or in the case of plant, this is a cell. Okay. In this cell, oxygen is entering. Your cell, oxygen is entering. Yes. The food is also entering the cell. This food is also entering the cell. You're eating food. The food is getting oxidized here. The food is getting oxidized, or oxidation of food is happening. That is producing energy. That is producing energy, and energy is stored in the form of ATP. This is the story of respiration. That is oxidation of food to produce energy, and the energy is stored in the form of ATP. This is the story of respiration. Okay, one second, friends. One second. Let me just check. Yes, this is the story of your respiration. Clear, all of you? Clear? Now, students, but what is the definition of respiration? What is the definition of respiration? Definition of respiration is very simple. Now, whatever food which you eat, right? Is it simple or complex? You tell me that first of all. It is complex carbohydrates, lipids, proteins. Yes. So, what is happening in respiration? In respiration, complex food, right? Complex food is getting converted to simple food, monosaccharides, amino acids, right? Fatty acids, simple food. Simple food. Complex food is getting converted to simple food. And what is happening here? The carbon-carbon bond is broken. In the process, carbon-carbon bond is broken here. Broken. How? With the help of oxidation. Through oxidation. And where is it happening? All is happening within the cell. Within the cell. And when the complex food, carbon-carbon bond is broken down in the what is called oxidation, converted into simple food in the presence of within the cell. This entire process will release energy. Energy stored in the form of ATP. This entire process is called as respiration. Entire process is called as respiration. Okay. Now, next question is, what is respiratory substrate? For example, here I told you that the food which I spoke about here, or the food which I'm talking about here, that food is nothing but the respiratory substrate. That's all. The food is called as the respiratory substrate. The food is called as respiratory substrate. But there are different types of food. Like how you have different type of biryani, different type of chapati, different type of paneer butter masala, chicken butter masala, dal makhani, different type of food. There is also different type of substrate which our body can take. For example, the first one is called as your glucose, the favorite one, the glucose. And this glucose is a type of carbohydrate. Glucose is a type of carbohydrate. Glucose is a type of carbohydrate. Then we have fats. Then we have proteins which can be taken by body, backup. At the same time, certain organic acids also can be taken. Organic acids can also be taken by your body. Yes, like malic acid and everything. This is called as your respiratory substrates. Substrates which can be broken down by the body to give energy. Clear? Simple. Now, what about vitamins? Vitamins, minerals, you know, minerals, water, hormones, all of the following cannot be respiratory substrates because they do not generate energy. Vitamins and minerals are basically cofactors. They help in reactions, hormones also play a major role, water also plays a major role, but they do not release energy. So these are not respiratory substrates. Respiratory substrates can be your carbohydrates, fats, proteins, organic acids can be your respiratory substrates. Now, friends, I'll ask you one more question here. Understand, carefully understand. Here I told you, complex substance is broken to simple substance. Does this definition sound very similar to something called as catabolism? Tell me yes or no. Does it sound very similar to catabolism? Complex substance is getting converted to simple substance. Catabolism, pure catabolism. Right. But students, for now, understand it is catabolism. But as we go towards towards the end of the chapter, you will learn a new process called as amphiboic process. That time we will see something different, magic. Okay. So right now, catabolism. Right now, catabolism. Now, students, in this process, energy is being produced or consumed. Tell me. You tell me, sir. This energy is being produced here. So can I also call it as exothermic reaction? Yes, I can also call it exothermic reaction. Exothermic reaction. And also physicochemical reaction. Remember all those points. Respiratory substance done. Now what is currency? Like all of you, I've been telling here from that time, ATP, here also ATP. What is this ATP, students? What is ATP? ATP is called as energy currency vessel. I'll give you a simple example. Okay. Back in the days, back in the days, I will give you wheat, you give me rice. Barter system used to work. Right. Later on, barter system was replaced by currency, gold coins, silver coins, bronze coins, currency notes, 10 rupees, 20 rupees notes. Now, every single time, right now, if you want to buy that wheat or rice or something, what do? Do you go take 1 kg chicken and give and they'll give you rice? No. You give the money, they give you the food. Here also, this ATP is just like your money. ATP is just like the money. You give the ATP, energy is given to you. Clear? But how is the ATP formed first of all? That is very important to understand. See students, first of all, we have something called as ADP. This is the ADP right here. And we have a phosphate here. This phosphate has to attach to this ADP. But attaching also requires energy. So, I told you, your body is producing energy, right? Your body is producing energy. This energy is consumed. Your body is generating energy, right? That energy is used to make this bond formation, and that's how we get the ATP. So ADP is converted to ATP by using the energy in the starting. So all the energy is produced right here. I told you, energy is produced here. Energy is also produced here. So all of this energy is used to make the bond and form the ATP. So if I ask you a question, where is the energy stored in ATP? You will tell me, sir, energy is stored in the bonds right here. Energy is stored in the bonds. Now you tell me, sir, if I need some, for example, I am going for marathon running. I need more energy. How do I get more energy now? So what happens here when your body requires energy? This ATP, they are adenosine triphosphate, and I told you energy is stored in the bond. So just, if I need energy, what do I have to do? If I need energy, what do I have to do? Break the bond. Simple. So this bond is broken. This bond is broken. Energy is released, and again it is converted back to ADP. Do you understand the concept? Every single time you have energy, use the energy and make ATP. Now, when you require the energy, break the ATP and take the energy back. Very simple. When you have a lot of money, you give your money to your mom or dad. They will keep the money for you. And every single time you need the money back, you ask your mom or dad, they'll give you back the money. But that is not the case in reality. Someone guest come to your house, guest will give the money will go to your mom and never come back to you. That is the untold story. I don't know why it always happens. Someone guest comes to your house, they give you money, you give it to your mother. Mother will be like, I will take care of this money, but the money will never come back to you. Sad reality, right? But body is very honest. Our body will make sure that every single time there is a requirement, ATP is broken down, you get ADP, and energy is given to you. Okay. Now, students, then what is the concept of NADH then? Understand. So all of you know ATP is like money. All of you know ATP is like money now. Now your NADH or FADH is something like check. Check. you have a check, for example, you have a 10,000 rupees check. Can you take the check and go to the bakery and give the check and tell them, I want 10 samosas, 10 kachori, 10 egg puffs? Will they give you? They will like, next, go to next shop. Why? Because they can't take the check directly. So what do? You take the check, you go to the bank, deposit the check, then get the money, then take the money, and then go to the bakery and buy whatever you want. He will take the money that time. Similarly, inside your body also, there is something called as NADH and FADH, which is just like a check. This check can be taken, and with the help of this check, we can get 3 ATP from NADH. From 1 FADH, we can get 2 ATP. You take the check, deposit the check, you get the money. Take NADH, deposit the NADH, you get the ATP currency. Simple concept. Understood, all of you? Understood? Beautiful. Now, friends, one more big question is that where do plants get the respiratory substrate from? Like I told you, we are eating food from the outside. I am going to the market, taking my ramen, taking my Maggi packet, taking my Coca-Cola, and coming back home, eating, drinking nicely, and enjoying. So I got my food from the outside. Or if mother is home, she's making nice food, nice batting I'm doing that day, eating nicely, I'm eating the chapati, palau, nicely I'm eating. So I am getting my food from somewhere. But where do you think plants are getting it from? Do plants also have a shopping market where they sell plant parts which plant can buy? Some secret shopping market which we don't know? That's not the case. Then from where do plants get the respiratory substrate? Are they also buying from somewhere? Friends, do you remember this class 6 definition? Plants make their own food? That is the answer. So plants do not have to buy from anywhere. Plants can make their own respiratory substrate, that is glucose. All of you know this equation from the previous chapter. Carbon dioxide plus water in the presence of sunlight and chlorophyll. Glucose plus oxygen. This glucose right here can be taken by the plants. Now, understand the difference between autotrophs and heterotrophs now, once and for all. If you know this much now, understand this difference, simple difference between autotrophs and heterotrophs. Understand the difference. For example, for autotrophs, this is the plant cell. Heterotrophs, this is the plant cell. What is happening in our case, heterotrophs? We are us movement right now. Okay. We are taking the oxygen in, we are taking the food in, and we are getting it oxidized. Oxidation. Oxidation is happening, producing the energy. Energy is converted to ATP. But in the case of plants, oxygen is entering, or oxygen is produced by the body itself, plant body itself, byproduct of your photosynthesis. Right here, you can see here, oxygen. Plants have it already. But in the case of plants, the food is not coming from the outside. In the case of plants, the food is not coming from the outside, especially for the green leaf, green parts. Especially for the green parts, the food is not coming from the outside. The food is prepared inside directly, and this food is oxidized to produce energy, and that energy is stored in the form of ATP. Simple difference. Autotrophs, plants are preparing the food inside the cell. Heterotrophs, it is coming from the outside. Very simple difference between autotrophs and heterotrophs. Now remember students, there are exceptions to this also. There are exceptions. Exceptions even in the case of plants. For example, roots. Obviously not, roots cannot make their own food. Flower cannot make their own food. Branches cannot make their own food. So in such plant parts also, there is heterotrophic mode of nutrition or transport of food is happening. Okay. So only in the green leaf, green stem, this scenario is happening. Where the food is prepared inside the cell itself. Okay. Now let's see the NCERT. What is given you NCERT? Let's drink some water. I hope the concept you're able to understand the concept here. Very simple. The chapter is very simple. All of us breathe to live. Obviously, if you don't breathe, you'll die. Simple as that. Why is breathing so essential to life? To survive. What happens when we breathe? Butterflies. Okay. Do all living organisms, including plants and microbes, also breathe? Yes, they also breathe. They might not have a nose or a mouth, but they also breathe. Okay. All living organisms need energy for carrying out daily activities like absorption, transport, movement, reproduction, or even breathing. So every single step requires energy. I told you that. Okay. Where does all the energy come from? Spoiler alert, food. Okay. Now, next question is, how is the energy utilized? Do all food have the same amount of energy? Obviously not. Some gives more energy, some will give less energy. Do plants eat? Yes, they also eat. but they make their own food. Where do where do plants get the energy from? And microbes of for their energy requirements. Do they also eat food? Yes, everyone eats food. Food is universal. You may wonder at several questions raised above. They may seem very discontinued, but in reality, the process of breathing is very much connected to process of releasing energy from the food. Because when you breathe in, you take oxygen. Oxygen is required for oxidation. Let us try to understand how this happens now. Okay. All the energy required for life process is obtained by oxidation of some macromolecules. By oxidation. What is oxidation? Loss of protons or loss of electrons. That we call food. Macromolecule food. Complex food. Only green plants and cyanobacteria can prepare their own food. Green algae. By the process of photosynthesis. They trap the light energy, light reaction, and convert it to chemical energy, dark reaction. That is stored in the bonds of the carbohydrate like glucose, sucrose, starch. We must remember that in green plants too, not all, very important, let's see, very important, not all cells, tissues, and organs photosynthesize. Only cells containing chloroplast, green parts, green stem, green leaf, that are the most often located in the superficial areas cannot photosynthesize. Hence, even in the green plants, all other organs, tissues, and cells that are non-green, root, stem, branches, flower, need food for oxidation. So they also need food. Hence, the food has to be translocated from all the non-green parts. Animals heterotrophic, that is, they obtain the food from the plants. Directly herbivorous, as they are omnivorous, but still. Indirect carnivores are non-veg cousins. Who are the non-veg cousins? That is a lion, tiger, and all. Saprophytes like fungi, one weird thing, fungi, are dependent on the dead and decaying matter, saprophytic mode of nutrition. What is the important to recognize that is ultimately all food that is required for life process comes from the photosynthesis primary source. This chapter deals with the cellular respiration or the mechanism of breakdown of food material within the cell. This is a definition of respiration. This chapter deals with cellular respiration or mechanism of breakdown of food material within the cell to release energy and trapping of this energy for synthesis of ATP. That is the entire chapter. Photosynthesis, of course, takes place within the chloroplast in the eukaryotes, whereas the breakdown of complex material to yield energy takes place in the cytoplasm and in mitochondria. Two important points here. Where does the respiration takes place? Where does the respiration takes place? What should be your answer? Cytoplasm, where both aerobic and anaerobic takes place, and also the mitochondria. Also the mitochondria. Two different things here. Okay. The breaking definition is here. Sorry. The breaking of CC bond, carbon-carbon bond of complex compounds through oxidation within the cell, leading to the release of considerable amount of energy, is called as the respiration. You will that here. You learned it here, right? Complex food, breaking of CC bond through oxidation, simple food within the cell to produce energy. Energy is stored in the form of ATP. We learned the same thing. Okay. Now, the compounds that are oxidized during process called as respiratory substrates. The food. Carbohydrates are oxidized to release energy, but the proteins, fats, and even organic acids are used as respiratory substrates in some plants under certain conditions. During oxidation within the cell, all the energy contained in the respiratory substrate is not released free into the cell. So all the energy is not released into the cell directly. The cell cannot take it from the energy. Okay, or in a single step, all the energy is not released in single step. Imagine instead of giving you one bottle of water, I'll give you one entire can directly to your mouth. Will you be able to drink it? No, sir. Give slowly. Water should be drink slowly. Should be drunken slowly, right? So even in the case of plants also, energy is released in a stepwise manner. You cannot release all the energy at one go only. The plant will not cannot take it. Okay. So it is realized that it is released in a series of slow stepwise reactions controlled by the enzymes, and it is trapped as chemical energy in the form of ATP. We learned that. And I'll give you a quick example here for stepwise reactions. The best example is glycolysis, 10-step reaction. Glycolysis is a 10-step reaction. Imagine that 10-step reaction, glycolysis can take place. Okay. Now, hence, it is important to understand that the energy released by oxidation in respiration is not, or rather cannot be used directly. So all the energy cannot be used directly, but is used as used to synthesize ATP. So all the energy is used to make ATP, and whenever there is a requirement, ATP is broken down, we get the energy back. Okay, when it broken down, and whenever energy is need to utilized, hence ATP acts as the energy currency of the cell. This energy is trapped in ATP is utilized in various energy requiring process of the organism. And the carbon skeleton produced during the respiration is used as a precursor for biosynthesis of other molecules in the cell. Now, what is the meaning of this last line? Very simple. In the entire respiration, there are a lot of intermediates like pyruvate, acetyl CoA, malic acid, DHAP, right? Alpha ketoglutarate. Now, all of these intermediates can be used in other reactions also. Okay, that is the meaning of the last line there. Now, respiration, we'll talk about aerobic respiration first of all. What is aerobic respiration? In aerobic respiration, there is oxygen is being used, first point, and lot of energy is produced. Second point is what? Lot of energy is produced. Lot of energy is produced. And it is a complete oxidation. What is the meaning of complete oxidation? Complete oxidation is that 6 carbon is used, 6 carbon is released. Equal, 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 equal. Right? So complete oxidation is happening here. But are there only one? No. There are two types of reactions mainly. There is something called as aerobic respiration and anaerobic respiration. What is the simple difference? Simple difference is in aerobic respiration, it takes place in the presence of oxygen. Oxygen is present here. Anaerobic, absence of oxygen. Very simple difference. First major difference between both of them. Aerobic respiration can take place in the cytoplasm, and also mitochondria. Also mitochondria. Anaerobic respiration only cytoplasm. Only and only cytoplasm. In aerobic respiration, lot of energy is produced. Lot of energy is produced. 30 ATP, 36 ATP. But in the case of anaerobic respiration, less energy, 2 ATP. Less energy is produced here. Less energy is produced in the case of anaerobic respiration. Clear? Now, under anaerobic respiration, there is something called as fermentation. Under anaerobic respiration, there is something called as fermentation, which is broadly of two types. Alcoholic fermentation, which happens in the case of yeast mainly, the beer, alcohol they make, and lactic acid fermentation, which takes place in the case of muscle cells or lactic acid bacteria. All of you know, on sports day, you will run nicely sports day. Next day, you will have a lot of muscle pain. That is because of lactic acid accumulation. Okay. Now, next question, students. Next question. Do plants breathe? Question is very simple. Do plants breathe? The answer should be technically, do they breathe? No. But do they require oxygen? Yes. Technically speaking, they don't breathe. The only exchange of gases is happening. But yeah. Well, the answer to this question is not quite so direct. Yes, the plants require O2 for respiration to occur, but they also give out CO2. Hence, plants have the system in place that ensures availability of oxygen. Some of the way plants will have oxygen. Plants, unlike animals, have no special organ. Obviously, we have nose, mouth, breathing, breathing. Plants do not have nose or mouth, so how do they breathe? Right? For gas exchange. But they have stomata. They have something called as lenticels on the stem or the bark for exchange of gases. In the case of plants, yes, they have lenticels. There are several reasons why plants can get along without respiratory organ. They don't require respiratory organ. The first one being what? The first one is, can you see? The first one is what? The plants can take care of themselves. They're not dependent on anyone. They can take care. They can take care of their oxygen requirement. They can take care of the they can take care of their oxygen requirements themselves. Easily they can take care of themselves. The second one is very simple. They do not have great demand. They don't require much of oxygen as us. There is no great demand at all. Plants can produce their own oxygen, use their own oxygen. They do not have great demand. Even if they have demand, how is it taken care of? Even if they have demand, remember, always remember, in the case of your photosynthesis, one of the byproduct of photosynthesis is because of photolysation of water is oxygen. O2 is byproduct. O2 is a byproduct in photosynthesis, yes or no? In photosynthesis. So can this O2 be used in respiration? Yes, it can be used. So not very demand. Now one more third main point is, if you look at the typical structure of a stem, the inner part is mostly dead. Inner part is mostly dead. Colon yes, or fibers. On the outside part, we have most living part is always on the outside. Living part is always on the outside. Always. Living part is always on the outside. So what exactly happens here is living part, which has chlorophyll and everything. If there is oxygen entering with the Vedantu cells, it can also do easy diffusion because all the living cells on the surface, all the living cells on the surface, so easy diffusion can happen and plants can take care of themselves. That's why they don't require very special organ. Okay, let's see the lines of NCERT now. First, each plant part can take care of its own gaseous needs. They can take care of their own requirements. Second, very little transport of gases from the other parts of other parts from other. Second, plants do not present great demand of gas exchange. No great demand. Okay. Root, stem, leaf prepare at respire very at lower rate than animals do. Only during photosynthesis, a large volume of gas is exchanged, and each leaf is well adapted to take care of its own needs. That is photosynthesis. When the cells photosynthesize, available O2 is not a problem in this cell since O2 is released within the cell. Second point right here, done. Third point, okay, the distance that gases must diffuse, even large bulky plant is not great. So all the diffusion can happen very close by, easily. Each cell is located quite close to the surface of the plant. This is true for leaves, you may ask. But what about thick woody stem? The living cells are surrounded by a thin layer inside the beneath the bark. They have a opening called as lenticels. So with the help of lenticels, they can breathe easily. These cells in the interior, the cells in the interior are dead and provide only mechanical support. I told you, inside is dead, outside is living. Thus, most of the cells of the plant have a least a part of the surface in contact with the air. This also is facilitated by the loose packing of parenchyma cells in the leaf, the stem, and the root, which provides interconnected network of airspace. So basically, telling you is all the living cells on the outside. You see here, all the living cells are on the outside here. All the living cells are on the outside. Easy diffusion can happen. Easy diffusion can take place here, and plants do not require much of tension. They also have lenticels. They also have lenticels. The complete combustion of glucose, which produces CO2 and water as an end product, yields energy, most of which is given as heat. Okay, this energy is useful to the cell. It should be available to synthesize other molecules that the cell requires. The strategy that the plant cells use is to catabolize the glucose molecule in such a way that energy is not liberated and liberated energy goes as heat. The basically telling you where students understand. Combustion and respiration are same to same. In combustion respiration, energy is produced. In combustion energy is used with the help of glucose or any other something like fuel, not glucose, any fuel. Oxygen is there, fire is burning, combustion. But the major difference between combustion and respiration is that all the energy is released in one go in combustion. But in respiration, all the energy is produced in a stepwise process. That is why there is no waste of energy in respiration. Okay. The key to oxidize glucose is not in one step, but several small steps enabling some steps to just large enough so that the energy released can be coupled to ATP synthesis. This is how this is how this how is this is done? Essentially, the story of respiration. Okay. During the process of respiration, glucose is utilized, all of you know, and carbon dioxide, water, and energy are released as products. The combustion requires oxygen, but some cells live where oxygen may or may not be available. Anaerobic conditions. There are sufficient reasons to believe that the first cell on the planet lived in an atmosphere that is lack oxygen because it was evolved in such a way. Even among the present day living organisms, we know of several that are adapted to anaerobic conditions. Bacteria, right? Some of these organisms are facultative, means flexible anaerobes, while some require anaerobic conditions in obligate. Obligate means strict, they strictly need it. Like how we are obligate aerobes, we need oxygen. Without oxygen, we cannot survive. There are certain organisms which does not require oxygen at all. Even if there is little bit of oxygen, they will die. Opposite of us, complete opposite of us. In any case, all living organisms retain enzymatic machinery to partially oxidize glucose without the help of glucose, right? Oxidize glucose without the help of oxygen. That is basically they're telling you is the first step in respiration, which is glycolysis, does not require oxygen. Simple as that. This glycolysis does not require oxygen. If it does not require oxygen, can I tell one thing? Glycolysis is a step which is common to aerobes and anaerobes. True or false? Tell me. True or false? True. So with that being said, now we will start the glycolysis. Now we'll start the glycolysis. Now friends, let's take a 10 minutes break and then we'll start glycolysis. I want everyone to refresh, right? I want everyone to refresh and we will start. Okay? 10 minutes break. Sorry, students, I had a sneeze coming up and I couldn't control it, so I had to go wash my face and come. Now, so that is the end of the introduction, right? We learned about so many things. We learned about what is the difference between your allotropes, heterotrophs, what is the meaning of respiration, how respiration is different from breathing, what exactly happens in respiration. That is, the food is entering, the food is getting oxidized to produce the energy. Now, students, the next step is called as glycolysis. Now, what is the meaning of glycolysis? How does it affect and what are the process in glycolysis? Let's understand. Okay. First of all, let me tell you what is glycolysis. Glycolysis is actually the first step in the respiration. It is the first step in the entire respiration. First step in the entire respiration is called as the glycolysis. Glycolysis. First step in respiration. Now, what is the meaning of glycolysis or glycolysis? Glyco means sugar. Glyco means sugar, and lysis means to break down. Break down. So what is glycolysis? Breaking of sugar is called as glycolysis. And this breaking of sugar can produce energy. Can produce energy. And this energy can be stored in the form of ATP. Can be stored in the form of adenosine triphosphate. Clear, all of you? Clear? Now, students, understand few things about glycolysis before we start glycolysis. There are few things which you need to understand about glycolysis. The first main point here is location of glycolysis. The first main point is location. Glycolysis takes place in the cytoplasm. In the cytoplasm of a particular cell. Of a cell. Cytoplasm of a cell. That is the location of glycolysis. Second point is the steps. Number of steps. There are in total of 10 steps in the glycolysis. 10 steps in entire glycolysis. 10 steps. Very good. The third main point is that where does it take place? In what organisms does it take place? In what organisms does it take place? Students, almost all organisms. All. It could be aerobic bacteria, anaerobic bacteria, prokaryotes, eukaryotes, animals, plants. In all of these organisms does it take place. But in the case of plants, it is special. Let me tell you why. Because in the case of plants, every single time the food is produced, the food is stored in the form of something called a starch. Starch. And this starch has to be converted back to glucose. See, plants make glucose. Plants make glucose. The glucose is stored as starch. Again, this starch is a complex polysaccharide, right? Or let me tell you, instead of starch, let's just call it sucrose. Instead of plants, I will just instead of starch, it's a little complex. Let's call it sucrose. Sucrose. Sucrose is a disaccharide. Sucrose is a disaccharide made up of your glucose and fructose. So the starch cannot enter glycolysis on its own. It cannot. So it has to again convert it back to glucose, and then only glucose can enter the glycolysis. This entire process enzyme mediated. The enzyme involved here is interkinase. Interkinase is the enzyme present here. Interkinase. Okay. So in the case of plants, first glucose is made. Glucose is stored in the form of sucrose. Sucrose is again converted back to glucose, and then only glucose is being used in glycolysis. Okay. Now, all of you know a few things I told in previous class also. What is the meaning of oxidation? What is the meaning of reduction? What is the meaning of oxidation? Loss of electrons or loss of proton, H plus, is called as oxidation. Then what is reduction? Reduction is nothing but gain of electrons. We are gaining the electrons or gaining the protons. That is called as your reduction. Gain of electrons or gain of proton is called as your reduction. Clear? Gain. I hope that is clear. Now, friends, your glycolysis can also be called as EMP pathway. Now they'll ask you why is it called as EMP pathway. Question is very simple. Answer is even more simple. It is called as EMP pathway because it is the initials, the first letter of the second name. For example, E from Gustav Emden, M from Otto Merov, P from Jacob Parnas, EMP pathway. Because these three were the scientists who discovered the glycolysis, and hence it is also called as EMP pathway. Clear? Now, the entire glycolysis, can be divided into two stages. Like how before you get your money, you always need to invest something, then only you get the money back. Without investing, will you get the money? For example, you're working, you have to work to get money. You're buying stock market, you have to invest the money, then you get the returns. Here also, you're investing the energy. In preparatory stage, you are investing the energy. In the payoff stage, you are getting the energy back. You are receiving the energy in the payoff stage. You are basically upload, download. Simple. Upload in the case of preparatory, download in the case of payoff in terms of energy. Simple. Now, what happens in preparatory stage? See, I've given very detailed. I've told you. On the right side, I'll be showing the entire your glycolysis. On the left side, we'll be doing the reactions. The first step is called as phosphorylation. The first step is called as phosphorylation. So all of you know glucose, which is a 1, 2, 3, 4, 5, 6. 6 carbon molecule, C6H12O6. 6 carbon molecule called as glucose. Now what is happening in the phosphorylation in the presence of enzyme hexokinase? Remember students, first enzyme, last enzyme, very important. Okay. In the presence of hexokinase, in the presence of hexokinase, right, in the presence of hexokinase, what is happening? Glucose is phosphorylated. Glucose is what? Phosphorylated, or phosphorylation is happening. In phosphorylation, we are adding a phosphate. Where are we getting this phosphate from? You see this ATP here? Yes, sir, we see the ATP. This bond is broken here. This bond is broken. When this bond is broken right here, this phosphate will be free. This phosphate, the free phosphate is going and it is attaching at the 6th position of the glucose. This phosphate is attaching to the 6th position of the glucose. So now I call it glucose is right here. So can I call it now glucose 6th position phosphate, glucose 6 phosphate? Yes or no? Tell me. Yes or no? So glucose becomes glucose 6 phosphate. Glucose gets converted to glucose 6 phosphate. That is the beauty of first stage called as preparatory stage. First stage. Okay. I'm gonna write this thing for you here so you can remember. First, second, third, fourth, fifth isomerization, sixth, seventh, eighth, ninth, and 10th. Clear? Clear of you? This is a 10th step. This is 7th step. So if I ask you in which step ATP is being consumed, you are supposed to say first step ATP is consumed for now. Later we'll add. First step ATP is being consumed here. Okay. Now this glucose 6 phosphate we have. Now obviously ATP is consumed, it will become ADP, it will become ADP. Now the second step is called as isomerization. Isomerization for the second step. Now there is something called as isomers. In chemistry you might have learned about isomers. Same but different. What is happening in isomer? In isomer, the chemical formula is same, but the structure is different. So for example, glucose 6 phosphate and fructose 6 phosphate, both formula is C6H11O9P. But structure wise, if you look, here we have an aldehyde group, here we have a ketone group. Structurally they are different. Chemically they are same. They're called as isomers. So in the second step, isomerization is happening in the second step. What is happening? Isomerization is happening in the second step. Glucose 6 phosphate to fructose 6 phosphate. And remember students, in isomerization, there is never release of energy or energy is never taken in. It is always neutral. Okay? That is Isomerization is happening in the second step. The third step, what is happening? Understand. Again, in the third step also, one ATP is here. This bond is broken right here. This phosphate is free. And this phosphate is added to the first carbon. Added to the first carbon. So before it was called as what? Fructose 6-phosphate. After glucose became fructose, isomerization. Fructose 6-phosphate, if I'm calling this, if I add one more phosphate here, now can I call it fructose 1, comma 6 bisphosphate? Yes or no? Tell me. Yes or no? Fructose 1, comma 6 bisphosphate. Third step. Done. Fructose 1, comma 6 bisphosphate. Done. Again, ATP is consumed here. So if I ask you the question, in which step ATP is being consumed, your answer should be first step and third step ATP is consumed. Now, what have we learned till now? Glucose to glucose 6-phosphate, ATP is consumed. Isomerization, then fructose to again one more ATP is consumed. Fructose 1, comma bisphosphate. Done. Till here we learned glucose, glucose phosphate, ATP is consumed. Isomerization, ATP is consumed again, and we end up getting fructose 1, comma 6 bisphosphate. Okay, fructose 1, comma 6 bisphosphate. Now, students, understand this fructose 1, comma 6 bisphosphate is how many carbon? So this is carbon here, phosphate, first carbon 2. So let me change the color, students. I hope it is visible. We have carbon, carbon, 3, 4, 5, 6. 6 carbon molecule. Here on phosphate, here on phosphate. 1, comma 6, 6 carbon. Now what if I tell you, if I take an axe and completely chop it off? 3 carbon on top, 3 carbon bottom. Do we end up getting two separate 3 carbon molecules now? Yes or no? We get two separate 3 carbon molecules. That is what is happening. And one 3 carbon molecule is called as glyceraldehyde 3-phosphate. One of them is called as what? Glyceraldehyde 3-phosphate. And the other one is called as what? Dihydroxyacetone phosphate. This is also called as your GAP, also called as PGAL. Same thing. PGAL. Glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde. Same meaning. This is called as DHAP. DHAP. That is, one 3 carbon molecule is here, one more 3 carbon molecule is here. 3. After cutting the in the middle. Now, friends, understand each one of them are also isomers. Now, if both of them are isomers, can I interchange them? Can I interchange them? Now, for convenience, I am interchanging DHAP into glyceraldehyde 3-phosphate. DHAP is converted to glyceraldehyde 3-phosphate. So we have what? Glyceraldehyde 3-phosphate right now we have. How many? Into 2. Why? Because one DHAP became glyceraldehyde 3-phosphate. So from this step onwards, every single place will add into 2 now. From here, into 2. So NCERT says here, triose phosphate, same name, both. GAP, DHAP. Triose means 3 carbon phosphate. Simple. 3 carbon phosphate compound, glyceraldehyde 3-phosphate. Now what do we have right now? Right now, this is the end of preparatory. Now we get the money back. Investment returns. That's happening in preparatory. In payoff now. Now what is happening in payoff? The first step in payoff is very simple. The first step in payoff, did we have the glyceraldehyde 3-phosphate into 2? Yes. This will undergo oxidation. Tell me, what is oxidation? Oxidation is nothing but loss of electrons. Loss of electrons or loss of protons. Loss of electrons or loss of protons. It is happening. Okay. Now it's undergoing oxidation. Electrons are produced, protons are produced, and also energy is produced. Now, what is the fate of these electrons and protons? Who will take them up? These electrons and protons are taken up by. These electrons and protons are taken up by your NAD plus. Nicotinamide adenine dinucleotide is taking the electrons and protons. Now you tell me, what do you call gain of electrons and protons now? It's gaining. Gain of electrons or gain of protons is called as reduction. So it is undergoing reduction to produce what? It is undergoing reduction to produce your NADH+. So this particular structure received the electrons, received protons, it undergoes reduction to produce NADH. And that is how in this step we get what? In this step we get 1 NADH. Now, what was this energy? What is the use of energy? This energy is now used. This energy is now used to bring one inorganic phosphate, one inorganic phosphate from the cytoplasm. And this energy is used to take this phosphate and attach it to the first position. And always remember, energy is stored where? Energy is always stored in the bonds. So energy is stored in the bonds here. So I'm adding one more phosphate here. So glycerol 3-phosphate, can I call it? It will become 1,3 something now. Wait, no. So what is happening in this step now? We are getting, look at the diagram of NCERT. Your DHAP got isomerized to PGA or gap. Okay, gap. This is getting, can you see one NADH here? Yes, one NADH we learned. It is getting converted to 1,3 bisphosphoglyceric acid. By 1,3 because one phosphate has been added here. 1,3 bisphosphoglyceric acid. That is BPGA. Normal PGA to BPGA. Normal PGA is getting converted to 2PGA. Okay. PGAL is getting converted to PGA. Okay. Always into 2 now. Always into 2. Now, students, energy is stored in the bond. Energy is stored in the bond. Now, what if this bond is unstable and this bond breaks and this phosphate is released? Energy can be released. Yes, energy can be released. That is what is happening. It is released. And this energy is used to take one ADP and this phosphate is added here. So can I tell ADP is now getting converted to ATP now? Can you see here? Glycerol 3-phosphate, 1,3 bisphosphoglyceric acid is converted to 3-phosphoglyceric acid because one phosphate is gone from here. And one ADP is produced where? In the first, second, third, 4, 5, 6 step NAD is produced, 7 step ATP is produced. 7 step ATP is produced. Otherwise, also 10 step ATP also produced in 10 step also. We'll get there. Okay, so ATP is produced in the 7th step. Now, 8th step, what is happening? Isomerization. This phosphate from here is going here. Simple. From here to shifting. Okay, and that time, this is happening. Okay, this step, 8th step. Okay, now in 9th step, what is happening? In 9th step, your 2-phosphoglyceric acid is losing water and getting converted to PEP phosphoenol pyruvate, which we learned in the C4 cycle. Which we learned in your C4 cycle, right? PEP phosphoenol pyruvate we're getting. Phosphoenol pyruvate. Okay, so what is being released out? What is being released out here? We have phosphoenol pyruvate into 2, everything into 2. Now again, this particular bond is unstable. It will break. This energy is taken and added to this phosphate here, and this ADP is now getting converted back to ATP. Here. Last step, 10th step, ATP is being produced here. This is your entire cycle of glycolysis. Okay, I'll give you a quick recap. Understand? Understand, friends. Understand. Understand. We'll be doing some water. I'll give you a quick recap. 10 minutes. Understand more time. Understand. Glycolysis. Glyco means sugar. Lysis means breakdown. Energy is produced to make ATP. First step in respiration, location is cytoplasm. 10th step happens in any of the following organisms. In the case of plants, sucrose is getting converted to glucose in the form of enzyme called as interkinase, and the entire thing is based on oxidation reduction. What is oxidation? Loss of electrons, reduction, gain of electrons. Done. It's also called as EMP pathway. Why? Because the discovery was done by 3 different scientists, Emden, Mayer of Parnas. There are 2 stages here. One is called as preparatory stage, other one is called as payoff stage. In preparatory stage, we invest, payoff, we take the return back. Okay. Preparatory stage, understand. Glucose is converted to glucose 6-phosphate, phosphorylation, ATP is added here. 6 carbon glucose, 1 phosphate attached to here will become glucose 6-phosphate. Glucose 6-phosphate. Now, glucose 6-phosphate will undergo isomerization, same but different. Second step, isomerization. So, we end up getting fructose 6-phosphate. Now, this fructose 6-phosphate again, phosphorylation is happening. Phosphorylation. So, can I tell this phosphate is added again here? ATP is broken down. This phosphate is added here. And fructose 6 phosphate will become fructose 1,6. 1,6 phosphate, bisphosphate. So till here is done. Till here is done. So ADP is used, ADP is used. In two steps, 1 and 3. 1 and 3. Now for fructose 5, fructose this is phosphate is getting converted to fructose 1, bisphosphate. We learned it. We learned it. Till here it is done. Now what is happening? Oxidation is happening. Right? Before oxidation, it is breaking down. It is breaking into two products. One is forming your visceraldehyde 3 phosphate, GAP or PGL, same name. Okay. Other one is DHAP, dihydroxyacetone phosphate. Now they are isomers again. Can they interchange? Yes, they can interchange. They can interchange. Here for convenience, DHAP is getting converted to visceraldehyde 3 phosphate. So that is why we have what? From fructose 6 phosphate, 1,6 bisphosphate, we get 2 molecules of visceraldehyde 3 phosphate. 2 molecules of visceraldehyde 3 phosphate. And now payoff starts. Now payoff starts. It's undergoing oxidation. What is oxidation? Loss of electrons and loss of protons. When it undergoes oxidation, it produces electrons and protons and energy. These electrons and protons are taken up by your NAD+. It is getting reduced to NADH. And that's how we get NADH in this reaction. Second thing, the energy is there. This energy is used to make a bond here with the help of a phosphate attachment. So visceraldehyde 3 phosphate will become 1,3 bisphosphoglyceric acid. 1,3 bisphosphoglyceric acid, and in this step, that is sixth step, NADH is produced here. Remember. Now this is undergoing what? Energy is broken down here. Energy is coming out. Energy is used to add this phosphate, and we get ADP. So 1,3 will become what? Just 3 phosphoglyceric acid, because one phosphate is lost here. This phosphate is lost. Okay. Now this phosphoglyceric acid is getting isomerized to 2-phosphoglycerate. 3-phosphoglyceric acid to 2-phosphoglycerate. This is traveling up, taking over and going up. Okay. 2-phosphoglycerate. The 2-phosphoglycerate is losing water to make the phosphoenol pyruvate. That is the 9th step. Question can ask, in which step water is lost? 9th step, water is lost. Okay. Now again the 10th step, what is happening? Same story. Bond is broken, phosphate is released, taken up by your ADP, getting converted to ATP. That is the 10th step. That is the end of it. Now the question is, what is the net profit here? Net profit of ATP. How much did we earn totally? Understand. Okay. In preparatory stage, understand preparatory stage. In preparatory stage, can I say -2 ATP? First step and third step, -2 ATP. In payoff stage, in the sixth step, we have something called as NADH formation, which will give you how many ATP? Which will give you 3 ATP. But it is always into 2 from here, into 2. So 3 into 2 is 6 ATP. 6 ATP. In the 7th step and 10th step, we get ATP. In the reaction, you can see only one. 2 basically. In the reaction, you can see 2 here. 1 here and 1 here. But everything is into 2 now. Into 2. From this step onwards. So totally how many ATP? 2 on top, 2 on bottom. So 4 ATP. 4 ATP right here. 4 plus 6 is 10 ATP. This is gross. Minus 2 ATP from the top. So we end up getting how many ATP? Net ATP is 8 ATP. This is the net. Net ATP is 10 ATP in the case of your glycolysis. Now the biggest question is, how can the question be asked? How can the question be asked? The first question they can ask, friends, understand this is very important. Understand this is very important, friends. Understand this is very important, okay? The first question they can ask you. The first question they can ask you is location. Is cytoplasm. Second question they can ask you is steps. 10 steps. Third question they can ask you is, which is the rate determining step? Means the slowest step. The rate determining step is going to be the third step. In the third step, where we are converting fructose 6 phosphate to fructose 1,6 bisphosphate. Third step, where ATP is also consumed. Next question they can ask you is irreversible steps. Irreversible steps. That irreversible steps is 1, 3, and 10. 1, 3, 10. Irreversible steps. Then they can ask you in step ATP is formed. That is 7 step. ATP is formed in 7th step and 10th step. Reactions are very important. Reactions are more important. In the 7th step, what is happening? In the 7th step, your 1,3 bisphosphonuric acid is converted to 3-phosphonuric acid. In the 10th step, phosphoenol pyruvate is getting converted to pyruvic acid. Pyruvic acid. In which step ATP is consumed? In the first and third. In which step water is produced? 9th step. 2-glycerate to phosphoenol pyruvate. In which step NAD is produced? 6th step. They can also ask you which step oxidation takes place. Okay, they can ask you so many questions. The first enzyme. The first enzyme, that is hexokinase. Hexokinase. Last enzyme. That is going to be pyruvate kinase. Pyruvate kinase. So all these questions they can ask you. All these questions they can ask you one by one. Okay. And this entire steps, basically this first step, these two steps, what is happening? Substrate level phosphorylation. Substrate level phosphorylation is happening in the 7th step and 10th step. 7th step and 10th step. Okay. Okay, students, all of these steps can be asked. And why they can also ask you EMP pathway. Why it's called EMP pathway. Okay. So all these questions can come from your glycolysis. Let's read NCERT now. Let's read NCERT. The term glycolysis originated by Greek word means glyco for sugar and lysis means splitting, remove. The scheme was given by 3 scientists, Gustav E. M. P. Parnas, often referred to as EMP pathway. In anaerobic organisms, it is the only process of respiration. Very important line. Only process of respiration in anaerobic organisms. Okay. Glycolysis occur in cytoplasm location and present in all living organisms, no exception. In this process, glucose undergoes partial oxidation. Very important line. Partial oxidation. Partial oxidation. Why? Because there is no CO2 is not evolved anywhere. Do you see anywhere CO2 evolved? No, CO2 is not evolved anywhere. So partial oxidation to form 2 molecules of pyruvic acid towards the end. In plants, glucose is derived from the sucrose, which is the end product of photosynthesis, or from the stored carbohydrate. Sucrose is converted to glucose and fructose by the enzyme invertase. We discussed this. By the 2 monosaccharides readily enter the glycolytic pathway. That is fructose and glucose. Glucose and fructose are phosphorylated by giving rise to from glucose 6 phosphate. First step. By the activity of the enzyme hexokinase, first enzyme. This phosphorylation often glucose then isomerized second step to produce fructose 6 phosphate. Subsequent steps metabolism of glucose and fructose are same. From here onwards, everything is same to same. The various steps in glycolysis depict in the figure. In glycolysis, the chain of 10 reactions, so 10 steps, under the control of different enzymes, takes place to produce the pyruvate from the glucose. While studying the 3 steps of glycolysis, please note the ATP being consumed anywhere. ATP is utilized in 2 steps. First step in the conversion of glucose to glucose phosphate, and the second time in the fructose to 6 phosphate to fructose 1,6 bisphosphate. Basically, the first step and third step in the reaction. Okay. Fructose 1,6 bisphosphate is split into DHAP and PGAL. Here, this step here, splitting into 2 things. We find there is one step where NADH is formed from NAD plus. This is when the 3-phosphoglyceric acid PGL is converted to BPGL. Okay. Two redox equivalents, H plus and electrons, are removed in the form of hydrogen atoms. Told you. And PgL is transformed into molecule of transferred to molecule of NAD plus. And PgL is oxidized. I told you, oxidize happening with inorganic phosphate to convert into BPGA. Oxidation is happening. The conversion of BPGA to 3-phosphoglyceric acid is also energy yielding process. So we are producing energy. Okay, we are also producing energy to form ATP. Here, this is the energy yielding process. Seventh step. Another ATP is synthesized during the conversion of PEP to phosphoryl acid. This one, 10th step. Pyruvic acid, when it is the key component of glycolysis, what is the metabolic fate of pyruvate? We will do that now. This depends on cellular needs. There are three major pathways. Okay, we will do this, students, in which the different cell handles pyruvic acid by glycolysis. Okay, that is fate of pyruvate. Now we'll take 15 minutes break and we'll come back and then we will do it. What is the time? Time right now is 2:06. Okay, let's take it as 2:05 only. We'll come at 2:10. 2:20 will start. Okay, plus resume at 2:20. Okay, so I'll see you at 2:20 p.m. over here. Bye bye. Now, students, now we will learn something called as the fate of pyruvate. Like, for example, what exactly happens to pyruvate after the glycolysis. Now, let's just write down first. One second. Okay. Mhm. Right here we have the first we have the glucose. We have the glucose right here. Glucose is getting converted to pyruvate. This process is called as what? Glycolysis. It takes place in the cytoplasm. In the cytoplasm. Okay. Now this pyruvate can undergo three different things now. This pyruvate can undergo three different things right now. Now, this pyruvate can undergo three different pathways. For example, first one is in the absence of oxygen. Second one is also in the absence of oxygen, in the absence of oxygen. And the third one is in the presence of oxygen. Presence of oxygen. Okay. The first one is happening near yeast. Second one is happening in the muscle cell. Muscle cell. And the third one is happening in the mitochondria. Happening in the mitochondria. Understand? So the one which is happening in yeast is called as your alcoholic fermentation. Alcoholic fermentation. In alcoholic fermentation, all of you know there is a release of ethanol plus CO2 plus water. In muscle cell, it is called as lactic acid fermentation. Here we have production of lactic acid plus water. Plus water. The third one is called as your aerobic respiration. Under your aerobic respiration, there is release of CO2 plus water plus energy. Here also plus energy. This is called as the fate of pyruvate. A pyruvate can undergo three different pathways depending upon where it is formed, or is there oxygen or oxygen is not there. Okay. Lactic acid fermentation, alcoholic fermentation, called as aerobic respiration. Now, the first one is called as your lactic acid fermentation, which happens in the case of muscle cells. Muscle cells. You went to you had to play sports day. Suddenly, there is lactic acid in your muscle cells. That's why you're not able to play really well. Okay, muscle cell. Now, what is happening here is understand, friends. This particular NADH is undergoing oxidation. It is undergoing what? Oxidation. Now, what is oxygen oxidation? Oxidation is loss of electrons, or can I call it loss of protons? Now, these electrons and these protons are taken up by the pyruvate. If pyruvate is receiving the electrons and protons, can one second, let me do this first here. NAD plus getting oxidized to NAD plus. It is pyruvate is accepting the electrons and accepting the protons. Can I tell you pyruvate is actually undergoing a reduction here? Reduction. And in terms of enzyme called as lactate dehydrogenase, that's all. You need to remember here the main point here is going to be pyruvate is the first electron acceptor. That's what lactic fermentation. What about alcoholic fermentation? In alcoholic fermentation, there are two steps. The first step is called as decarboxylation, enzyme pyruvate decarboxylase enzyme. And from 3 carbon will becoming 2 carbon, 1 C carbon is released. That's why they tell In the alcoholic fermentation, we have what? In alcoholic fermentation, we have ethanol plus CO2 plus your energy. Plus energy. So CO2 is released, yes, CO2 is released here. In the making cheese, CO2 is released out. In making dosa batter, CO2 is coming out. So we have acetaldehyde here, acetaldehyde. Okay. Now this NAD plus again undergoes oxidation to produce NAD plus. Electrons and protons are released. These electrons and protons are taken up by your acetaldehyde. If acetaldehyde is receiving the electrons and protons, it is undergoing oxidation. It is undergoing oxidation to produce the ethanol finally. Oxidation is undergoing, sorry, it is undergoing reduction here. 1 second. It is gaining electrons and protons. It is gaining electrons and protons. It is going reduction. Undergoing reduction. Here we have oxidation. And this acetaldehyde is the first electron acceptor in alcoholic fermentation. Enzyme alcohol dehydrogenase, pyruvate decarboxylase. Two different enzymes are here. Okay. Fermentation. Let's see what's given in NCERT. Let's see what is given in NCERT. In fermentation, say by yeast, the incomplete oxidation of glucose is achieved under the anaerobic conditions by a set of reactions where pyruvate acid is converted to CO2, all of you know, and ethanol, alcoholic fermentation, right? In the pyruvic acid and ethanol, alcoholic fermentation. Enzyme pyruvic acid decarboxylase and alcoholic dehydrogenase, two enzymes in alcoholic fermentation. The other organisms like bacteria produces lactic acid and from pyruvic acid. Bacteria and also muscle cells. The steps involved in the shows in the animal cell also like muscle cell during exercise where oxygen is inadequate for the cellular respiration. It is reduced to lactic acid. See, reduction. Reduced to lactic acid reduction by dehydrogenase enzyme, lactic dehydrogenase enzyme. The reduction agent in both is NADH, reduction agent, which is reoxidized to NAD plus. It is reoxidized to NAD plus. Clear? So what is happening here? Glucose to glyceraldehyde 3-phosphate, 3-phosphoglyceric acid, phosphoryl pyruvate, PEP. Till here it is glycolysis. Now here pyruvic acid can undergo lactic acid fermentation in the case of your muscle cells or bacteria, alcoholic fermentation in the case of yeast. In both the cases, whatever NADH is produced here, it is used here. Can I tell it is used here? That is why NCERT is saying reoxidized. So whatever NADH is produced here, it is being used again by your NADH here. Reusing is happening. Okay, reuse is happening. So NADH is getting converted back to NAD plus. Now you tell me the net ATP here. Net ATP. In glycolysis, totally how many ATP then? In preparatory stage, we can tell in glycolysis preparatory stage -2 ATP. Now since NADH is being consumed here, there is no NADH only. We cannot tell NADH now. Let's just write. Later on we can cancel it out. Okay. In payoff stage, we have NADH 2. We also have 4 ATP produced. Now in the pyruvic acid, in the fermentation, in both the fermentation, one NADH is also used. NADH is used up here. Can you see use? So can I cancel both of them? Whatever was here is used here. So cancel it out. So what is the ATP now again? 4 ATP here, -2 ATP here. 4 minus 2 is 2 ATP. So 2 ATP is the net gain here. This is 2 ATP in the entire cycle. Okay. See, in both lactic acid and alcoholic fermentation, not much energy is released. Less than 7% of the energies in glucose is released. Less than 7%. 2 ATP. And not all is trapped as high energy bond as ATP. Also, the process are hazardous. Either acid or alcohol is produced. Net ATP we told already. Right. When one molecule of glucose is fermented to alcohol or lactic acid, yeast poison themselves to death when the concentration of alcohol is more than 13%. So if it goes more than 13, yeast will die off. So it will maintain. 13% is maintained. Then how do we get maximum concentration of alcohol in beverages that are naturally fermented? Naturally fermented maximum is 13%. All the, if you want anything concentrated, you want to go distillation for anything concentrated version. Okay. Hmm. Then what is the process by which organism can carry out complete oxidation? That is aerobic respiration, complete oxidation. Okay. And where does it take place? Aerobic respiration takes place in the mitochondria. Takes place in the mitochondria. So anaerobic is done. Aerobic we will do in the next class. What is happening in aerobic respiration? Glucose plus oxygen, energy is released, a lot of energy is produced, carbon dioxide plus water. Now what is happening here? I'll give you a small preview of what we're learning in next class. We have cytoplasm here. We have pyruvate here. Pyruvate is produced in cytoplasm. This pyruvate will now be transported into the mitochondria. Transported into the mitochondria. Can you see here? And this pyruvate will undergo three different reactions in aerobic respiration. Three different reactions. One is your oxidative carboxylation. Second one is Krebs cycle. And the third one is oxidative phosphorylation. That is basically your electron transport chain. Three different reactions in the next step. Aerobic respiration. Your oxidative carboxylation in mitochondria, Krebs cycle, it's a mitochondrial matrix, Krebs cycle, and cristae, that is inner mitochondrial membrane, inner mitochondrial membrane for your electron transport chain in the next step. Okay. So students, we will stop there now. Next class we will be doing aerobic respiration, all the three in the next class itself. Okay. Thank you so much, students, for today's class. Have a great day ahead. Okay.